Hi, my name is Patrycja Krawczuk, and this summer I worked on a project called Automated Indexing of Bioinformatics Tools. During this presentation, I will discuss the goals and motivation for the project. I will highlight some of the methods I used during the implementation phase, as well as introduce you to the final product of the project, a platform called BioToolBay. Let's start with the motivation. The recent advancements in high-throughput technologies in fields such as genomics and transcriptomics have caused the influx of massive amounts of new biological information. This abundance of data has subsequently led to developments of novel computational methods to analyze and extract knowledge from these large datasets. Currently, every week, major journals in field of bioinformatics feature multiple publications about new software, databases, and other resources that assist biological research. However, after the publication date, these resources become increasingly difficult to locate. This is why we decided to create an automated and self-maintaining index of bioinformatics tools. Our goal was to build a platform that will enable researchers to find and discover new tools. Moreover, this project promotes Force 11 Community's Fairness Initiative by featuring Fair Shake status insignia developed by Lily and Dr. Avi Mayan. The initial step of the project required collecting data from biomedical literature. We decided to first index 4 major bioinformatics journals and gather all the papers published between 2000 and 2017, which in aggregate was over 40,000 articles. The information extracted included the publication's abstract, authors, number of citations, and more. Two Python libraries were important to the data analysis step. First, NLTK was used to process the abstracts from the publications about the collected tools. This slide shows an abstract of the article regarding Link's Canvas browser. The original passage has length of 196 words. However, during the pre-processing steps, such as removal of stop words, annotation of the remaining text tokens with part-of-speech tags, and laminization of words, this passage was distilled to 74 essential words. The second library, scikit-learn, was used to construct weighted feature vectors for all of the preprocessed abstracts. I decided to use the term frequency, inverse document frequency metric to reflect the importance of a given word in the accumulation of all the features in the collection. TF-IDF is a product of two metrics, term frequency and inverse document frequency. Its value increases proportionally to the number of times a specific word appears in a given article, but it is offset by the frequency of the word in the corpus. This helps to adjust weights for words that are used frequently throughout a given corpus. This slide shows a vector of the weighted features for Link's Canvas browser. On the right, we see the same vector visualized as a word cloud. For Link's Canvas browser, the algorithm considers words like expression, links, signature, and technology to be the most meaningful. To determine relationships between the tools in the collection, I applied cosine similarity. Two abstracts are considered similar if the angle between their feature vectors is approaching zero. To validate the results of the analysis, I created a 3D interactive visualization of the network of similar tools. This slide displays a snapshot of that visualization. The color scale represents the average number of citations per year, while the size of a ball corresponds to the scaled average number of views per year for that tool. The visible clusters indeed represent tools in the same domain having similar functions. All of these results are featured on the BioToolBay website, which currently hosts almost 6,000 unique tools, including packages, visualization tools, databases, algorithms, simulations, and more. To make the tools findable, we developed a user-friendly search interface that supports search by keyword and provides different filtering and sorting options. User can specify the type of the tool, narrow their results to those published in a chosen journal, or add additional keyword to the query. The results can then be sorted by publication date, popularity, or number of citations. This is an example of a specific tool landing page. Enricher is a tool developed by Mayan Lab. The landing page template includes an abstract from the publication of the tool, list of authors, metrics like number of views, citations, Almetric score, as well as fairness score retrieved through Fairshake API. Every landing page includes URL to tool's homepage or GitHub repository. One of the most important features of the page is the Similar Tools recommendation carousel that aims to provide users with 9 most similar tool suggestions. There are many opportunities to improve the functionality of BioToolBay website. One of them would be expansion of database to include other journals and repositories. I would like to also expand the querying options to make the search results more relevant. I would like to thank Dr. Avi Mayan for his guidance and his many great ideas. Denis Torre for his advice and patience, Lily Wang for adding Fairshake status insignia to BioToolBay, all members of the lab and fellow summer students for this memorable experience. Thank you.